In this video, we'll discuss about spread of tumor, that is metastasis. Here, we'll discussing three type of spread of tumor. The three route of spread of tumor: lymphatic, haematogenous, and transcoelomic. So, three spread of tumor, three-way spread of tumor: lymphatic, haematogenous, and transcoelomic. Let's start before coming on the route. Define the metastasis. How define this metastasis? What is metastasis? Metastasis is distance spread. So spread of tumor by inversion in such a way that there is discontinuous multiply secondary tumor or masses are formed at a site of lodgement away from the primary site, that the world to be pickled up here away. So spread of tumor by inversion in such a way that discontinuous secondary tumor masses are formed at the site of lodgement away from primary site. This is human body. This is the patient. Just supposed here there is the primary tumor. Primary tumor in any organ, in the abdomen, in the GIT. This is the primary tumor. The cells of the primary tumor will dislodged. Some of the cell and they will enter either in lymphatic or either in the blood vessels. Some of the tumor whether it will enter lymphatic or blood vessels or through that cavity only. They will transmit three-way transmission. One is through lymphatic or blood vessels or cavity. They will transmit to multiply secondary organ, and multiply secondaries can be formed. It may be affected in our brain, bone, in the liver, in the lungs, in the multiple organ. So now we want to notice multiple secondaries are there. All these secondary is or away from that primary tumor. So this one is the primary tumor. Now it will causes tumor in bone, brain, liver, lungs. So that one is the secondary tumor. So the word to be picked here. Okay, so from one place to another. Picked here discontinuous and away. They are discontinuous, they are away, they are not in continuation. If they are in continuation, it is known as local inversion, but since they are discontinuous, it is known as metastasis. So the other name of that metastasis is distance spread. So it is discontinuous, multiple secondary tumor and masses are formed at the site of lodgement, which is away and discontinuous from the primary tumor. Now metastasis is the most reliable picture of malignancy. There are many difference between benign tumor and a malignant tumor. We know there are the two type of the tumor, benign and malignant. The most reliable and short picture to differentiate between malignant and benign is metastasis. Benign tumor never show metastasis. Malignant tumor can show metastasis. So it is the short or the most reliable or the hallmark features of the malignant tumor, not benign tumor. So as benign tumor never metastasis. All malignant tumor shows metastasis except two. So benign tumor never metastasis. All malignant tumors show metastasis except two. First one is gliomas of CNS, gliomas of CNS, gloma of the brain, and the second is the basal cell carcinoma of the skin. Basal cell carcinoma of the skin. These two things shows metastasis. So these are the two tumor which are malignant. So ideally they should metastasis, but we have not seen single case of metastasis of these two, but looking at that morphology they are not benign, they are malignant. So the gliomas and basal cell carcinoma are the two tumor which are malignant, but they never shows metastasis. There are the exception. So point is that all benign tumor not show metastasis. All malignant shows metastasis except glioma and basal cell carcinoma. So what are the root of metastasis? There are three root of metastasis, that lymphatic, that haematogenous, and this transcoelomic spread. Okay, these are the three root of metastasis. Whether lymphatic, haematogenous means blood spread, then transcoelomic, through cavity it can spread. But this one is the rare, predominant lymphatic and haematogenous. So what are the root of metastasis? We discussed three. First root is lymphatic. We want to look at that lymphatic. This one is the rough sketch diagram of human body, female body, and she is having tumor in one of that breast. Now what are the way it can spread to the other organ? Or it can do the metastasis. Either the tumor cells can spread via lymphatic, or it can spread via blood vessel, whether it can spread lymphatic or blood vessels, or it can spread via transcoelomic route. Okay, what are the three route? So either the tumor cells can spread all other organ via lymphatic, or it can spread this one blood vessels, that is haematogenous route, and spread to the other organ. They shed directly in the pleural cavity. Okay, so they shed directly in the pleural cavity, the abdominal cavity, and via cavity it will moving to other organs. This is transcoelomic route. So lymphatic, blood vessels, transcoelomic route means through cavity it can transmitted. So these are the three route. So tumor cell will dislodge from the primary tumor and spread to that multiple secondary organ. What is it? Lymphatic, haematogenous, or transcoelomic? That are the three route. So we can see here in this tumor there is the tumor cell. This one is the tumor cell. The tumor cell will dislodge. They will enter either lymphatic, or they will enter through blood vessels. Okay, this one is the haematogenous. This one through lymphatic means that one is the lymphatic spread, and they will here also will spread to that multiple organ. So it will spread multiple organ. We know there are the two type of malignant tumor in human body, carcinoma and sarcoma. Carcinoma and sarcoma are the tumor of that epithelial origin. Okay, and sarcomas are the tumor of that mesenchymal origin. Tumor of epithelial origin, carcinoma. Sarcoma is the tumor of mesenchymal origin. So carcinoma spread via lymphatic route. So this lymphatic route will spread carcinoma. Carcinoma. This through blood. This one is the mesenchymal spread. Sarcoma or the tumor of mesenchymal origin. So carcinoma spread lymphatic route. Sarcoma spread hematogenous. Sarcoma. That is the thing. Now we will see the detail of them. The first is that lymphatic spread. Carcinoma spread by lymphatic route. What is the pattern? So the natural route of that lymphatic drainage that particular organ and producing regional nodal metastasis is the most common. For example, the breast, C, regional lymphatic or axillary. So axillary are involved. For the lung, the regional lymph node are paratracheal and perihilar. So that are involved. So the regional lymph node of that particular organ are involved. So someone is having breast cancer, the nodules which are involved are axillary. If someone is having lung cancer, the nodules which are involved are paratracheal and perihilar. So the regional lymph node are involved, that are the first thing. There are many lymph node, one by one they will be coming in the vein. So this one is the lymphatic vessel. One by one is the lymph node. This one is the lymph node are coming. This is the lymphatic vessels. This one is the lymphatic vessels. This one is the tumor. This one is the primary tumor. The primary tumor will come and the first node. And the first node they are involved all the nodes. Okay, the tumor involved in all the nodes which are coming in the way, the natural way. This one is the natural way. So all the lymph node will be involved one by one, but the first lymph node which is involved, it is known as sentinel lymph node. Okay, first lymph node involved, no, this name is called as sentinel lymph node. So the sentinel lymph node is of greater importance because by checking the sentinel lymph node we can predict whether the other lymph node are involved or not. If the first lymph node, the sentinel involved, there is a probability all of them ahead are also involved. But if the first node is not involved, if that sentinel lymph node is not involved, so there is less chances or no chances that the further lymph node are involved, especially in the breast cancer. So surgeon will take that primary tumor out of that breast either by performing lumpectomy or mastectomy. But whether axillary lymph node have to be taken out, yes or no, completely that depend on the sentinel lymph node. So During frozen section, we can test the sentinel lymph node is trophy 1. If the sentinel lymph node is involved, the lymph node should be also be removed. If that sentinel lymph node is not involved, lymph node should not be removed to prevent the further morbidity. So that is the relevance of that sentinel lymph node. So sentinel lymph node is the first node in that regional lymphatic is known as sentinel lymph node. It is a greater important in breast cancer, vulvar cancer and melanoma. It was the discovered by person is known as gout. Vulvar carcinoma and melanoma discovered the person is known as gout. See here, this one is the primary tumor. So if that lymph node are following this route, this is the first one. This one is the sentinel lymph node, and if the lymphatic are following this route, this is the sentinel lymph node. So the first lymph node is coming in the root is the sentinel lymph node. Usually the tumor cell involved in that lymph node in a sequence. The sequence is the natural sequence only. So this one is the natural sequence. So in the sequence, the first lymph node which is coming in the way that is involved first, then second, third, fourth, so on. But rarely from first directly to third. From first to third. It is an example of skip metastasis. This one is the skip metastasis. So rarely if there is an obstruction in the main pathway, there can be a skip metastasis by collateral takes place. It is very rare, but it can happen. So skip metastasis, sometimes the local lymph node may be bypassed because of various lymphatic anastomosis. The skip anastomosis, the skip metastasis can takes place now in a lymph node. The structure of that lymph node, lymph node is that capsules. This one is the capsule. Just flow the capsule follicles. Okay, here follicles are there, and the follicles are formed by that lymphatic. Follicles are formed by lymphocytes. The lymphocytes arranged themselves in a circular pattern from the follicles. So which portions of that lymph node is involved first by tumor cells? Okay, this one formed by tumor cells. The lymph node is involved first by the tumor cells. So answer of subcapsular sinus. This one is the capsule, this one is the follicles. This one here we have that cancer cell. Subcapsular sinus, here we have that cancer cells. So these all are the important point in that lymphatic. So what are the things this lymphatic? The first point, carcinoma spread by lymphatic. After that, it is the sequence. In the sequence, usually it is involved in regional lymph node are involved. Sometimes, rarely, there is a skip metastasis. Sometimes, really in lymph node, the subcapsular space in the first involvement. So here three things are important. First one is whether it can spread via sequence or skip metastasis or subcapsular. Okay, subcapsular space. Here tumor will form 3 places. Now coming to hematogenous spread. Hematogenous, that is the blood spread. So sarcoma spread by hematogenous spread by hematogenous spread. Sarcomas usually spread, not the carcinoma. Carcinoma spread by lymphatic. Now in the blood vessels. So this is the primary tumor. This is the vein. This one is the artery. What is the preference of the tumor cells? Will they spread via vein to other organ, or will they spread via artery to the other organ? What is the preference of the tumor cell? The preference is the vein. So tumor cell prefer to enter vein and spread to multiple organ and comparing to artery. So venous involvement of via venous, the spread is more common as compared to artery. The reason is very simple. Vein have a thin wall. We all know vein have a thin wall, but the artery have a thick wall. So tumor cell will get a resistance in the artery, but in the vein they can easily penetrate. So leaving the primary tumor, they enter that vein very easily as compared to artery, because artery have thick wall. That is hematogenous spread, the first point. The second thing, which organ are involved? So most commonly liver. Most commonly liver, followed by lungs. There are two most common organ involved because of that venous invasion. Hematogenous spread. Why liver is most commonly involved? Because of that portal drainage. What do you mean by portal drainage? The blood of complete GIT drain in that portal vein via portal vein only. It drain in that inferior vena cava. So the complete GIT, the stomach, the spleen, that pancreas, the small intestine, the large intestine, they all first drain in portal vein. Via portal vein it goes to liver. From liver it goes to SVC and IVC. They will go to IVC. So portal vein. Why? So if there is a malignancy in any of the GIT, if there is a malignancy, just suppose in stomach or spleen or in pancreas or in intestine or in GIT, so tumor cell will dislodge. They will go in the corresponding vein. From that corresponding vein they will go to portal vein. From portal vein the first metastasis in that liver. That is why liver is most commonly involved organ in the hematogenous spread, especially in GIT malignancy because of portal circulation. This is the reason. And the second is lungs. The second most commonly involved. Now apart from GIT, if tumor occur in any other organ of human body, any other organ, this is the tumor. Any organ, here tumor. Just suppose tumor cell will dislodge and they will go vein. So they will go to that vein of human body. They go in SVC, IVC. They will go right side of that heart. First they will go to right article, then they will go to right ventricle. From right ventricle they will go to pulmonary artery, and they will go to lungs. From the secondary, so okay, from lungs is the secondary one. So tumor is in any organ if it is spreading by haematogenous route, if it is spreading by vein, lungs. So the first organ which is coming in the way, lungs. So the second most commonly formed in the lungs, secondary most commonly formed in second organ in the haematogenous, most commonly it formed in the liver, second formed in lungs. So these are the two most common organ involvement of that haematogenous spread. Then various organ also affected. Apart from these lungs and liver, liver is the most common, lungs is the second most common. Apart from brain can be involved, bone can be involved, kidney, adrenal can be involved. Multiple secondaries are there. Why haematogenous route? Blood is going everywhere in body, so via blood they can reach any organ, but technically most commonly in GIT tumor will go in the liver, and other tumor will go in the lungs. So liver followed by lungs are the two most common organ involved in haematogenous spread. Now coming to third one is transcoelomic spread. What is transcoelomic spread? There are the three possibility in transcoelomic spread. Three possibilities through the blood cavity. The best example of body cavity is Krugenberg tumor. The best example of body cavities, Krugenberg tumor. See this diagram, Krugenberg tumor. They spread through body cavity. That is the one of the transcoelomic route. This is GIT. This is the stomach intestine of that female. There is a ovaries. This one is the ovaries. Here stomach GIT. This one is the ovaries of the female. Imagine this female is having stomach cancer. This lady having stomach cancer. That is the primary cancer. The gastric carcinoma, that is the primary. Now what is happening? The tumor cell are shedding in the cavity. So this one shedding in that cavity, it is not going via blood route, it is not going via lymphatic route, it is shedding in the cavity via floating in that cavity. Okay, in that floating in that cavity, it's involving that bilateral ovaries. In that bilateral ovaries, it is the secondary formation, it is the secondary. Okay, this one is the secondary. This is the primary one, these are the secondary one. Primary one is the stomach, secondary one is the ovary. So this is the triads, this one is the triads, is known as Krugenberg tumor. So basically patient present with three tumor, one in stomach, two in ovaries. The stomach one is the primary and the two ovaries are the secondary. The root of the spread, not blood, not lymphatic. Okay, this direct cavity, that is known as transcoelomic route. So why are cavities it's there? The best example, it through the natural field. The pleural cavity, Krugenberg tumor. From the stomach, the cells are going and involving the ovaries. So bilateral ovaries are very big. So basically the carcinoma of the stomach shedding into multiple or both ovaries. The secondaries are formed in the both ovaries. So it is a triad. The first thing in that transcoelomic route, the second thing spread via CSF. The CSF is presented in subarachnoid. So the CSF is presented in subarachnoid space in that brain and the spinal cord. So this is the brain, this is the spinal cord. In the this is the central nervous system. Now we have three managers are presented around the brain and the spinal cord. One of them is arachnoid and pyometer, and then arachnoid, that is durameter. So three things: arachnoid, pyometer, and durameter. In this subarachnoid space, that CSF is present. Subarachnoid CSF is present everywhere. Okay, this one is the CSF. The CSF is presented everywhere. The CSF is in that subarachnoid space. Suppose tumor is present in any portion of CNS. Tumor is presented in any portion of this CNS. Tumor cell will be shed in that CSF. Okay, via CSF it will go some other portion of that CNS only and form secondary. In the CNS, through that CSF only, it will transmit one place to another. Now define it is discontinuous. The second masses are discontinuous from each other, and the secondary is formed at that point of that lodgement which is away from primary. It is a metastasis. And what is the root? It is CSF. So CSF will also come in this transcoelomic route. So the tumor of that ephantema and leptomeninges can spread via CSF. That is the second things. Third things is implantation. Implantation is the rare of that rarest. For example, there is a surgeon, the surgeon using scalpel. Okay, surgeon using a scalpel or needle or suture in one of that cancer patient, and at one site of that the cancer patient, without sterilizing or without cleaning the scalpel, that same scalpel is used to cut some other site in that patient body. So surgeon scalpel is transmitting the cancer cell from one portion to another, one human to another. So that can be implantation. That is very rare. So what are the three root? We studied lymphatic haematogenous transcoelomic. In this transcoelomic we have three. First one is through cavity, Krugenberg through cavity. Second one is that CSF. Third one is implantation via surgeon scalpel. These are the three route. Now we want to discuss mechanism. Mechanism of metastasis. Mechanism of hematogenous root which is seen in sarcoma. There are 8 steps. Two steps in each diagram. So totally we have 4 diagram. First step is clonal proliferation and angiogenesis. First we understand this diagram. This is the tumor cell. This one is the blood vessels. Between tumor and blood vessels we can see extra tissues. Okay, we can see extra tissues. It is extravascular tissue. Okay, so the tumor cell first cluster of tumor cell will loosen. Cluster of tumor cell will loosen first. From the tumor cell, a cluster of tumor cell will become more aggressive. So more aggressive, cluster of cell more aggressive. That is the step number 1. So what is the step number 1? The heterogeneity. From the tumor, the cluster of tumor fuse cell become aggressive and they lose fusel, become aggressive and they separate, they become more aggressive, then they lose from rest of the tumor. For example, these 2 tumor cells or 3 tumor cells, they become aggressive, so they will lose. So what do mean by losing? What is losing means what is glue? What is holding the cell together? So for holding the cell together there is protein required. Okay, so it required a protein is known as E, stand for epithelial Catherine. Epithelial Catherine. So there is a loss of E Catherine takes place. So here loss of E Catherine takes place. So mutation in E Catherine, loss of E Catherine. E Catherine is acting like a glue. If there is loss of E Catherine, the tumor cell loosen from each other. After losing, they enter extravascular mass. So they will enter extravascular mass. EVM, the yellow color one. This yellow color one, extravascular mass. So we can see the tumor cell here. So first steps become the tumor cell become aggressive, which become loosened. They interacted with the extra that ECM, extracellular matrix, with the help of receptor. That receptor here, with the help of receptor. Receptors are laminin, pyronectin, vetronectin and collagen. So what are the receptor? Laminin, fibronectin, vetronectin and collagen. So the receptor are presented here via receptor. First they interact and after entering they degrade that extracellular matrix. they degrade extracellular matrix, they enter inside. So they enter inside. So they enter inside them for degrading. They have the enzyme collagenase and they having enzyme collagenase and gelatinase. And finally they enter inside extracellular matrix. After entry the extracellular matrix, they secrete autocrine motility factors. So after that they secreted autocrine motility factor via which it enter inside that blood vessels. So now it will enter inside the blood vessels. So it enter that lumen of that blood vessels and they rotate from here and there. They flow freely. So they flow freely from here and there. But what if the tumor cell or the foreign? Why not WBC kill them? Don't kill them because they are very small. They will form thrombus and covered themselves with RBC and platelet. Why our WBC not kill that tumor cell means this one is the small. So this they forming that thrombus. In this one covered by our RBC and platelet. So circulating host cell to prevent from that immune attack. Okay, that circulating host cell, that RBC and platelet prevented from WBC. So WBC cannot attack. That is the thrombus formation at the distant site. They will go out again. So they will go out again. This is known as extravasation. So this process name is called as extravasation. Extravasation, and they will form a secondary. They will do survival and they will produce a growth factor. They will do proliferation and they will form secondary here. So they will form secondary here. So these are the 8 steps. First cell, few tumor cell started aggressive. Step number 2, they lost from each other. For loosening, there is loss of loss of E catherine. Then number 3 interacted with the extracellular matrix. Number 4 enter inside extracellular matrix by degrading it. Step 5 they enter inside blood vessels through autocrine motility factor. Step 6 they form thrombus to protected from immune attack, protected from WBC attack. Then they extravasate at the some distant site, then they grow here. This is all about metastasis mechanism.